So, all right. We've already talked about bacteria, which is why we should be able to get through this quickly, which also means, again, if I'm getting rambly, tell me just to stop being rambly.
Get back on track.
Yeah. By the way, can I ask something? Because I can go either way. Do you guys find the little recaps at the beginning with questions helpful? Is that helpful to kind of cement your thinking and review things and keep it going? Because I've had classes in the past who say, please stop doing that. We'd rather just have a bit of time to work at the end. And I've had most classes say, like, yes, we find that to kind of be a useful thing to help remind us what we've learned thus far. I really am open to either way. You can kind of let me know. Oh, can I? Yeah, that's fine.
No, we cannot.
Nobody puts Joe in a corner, but here I am. How's that?
That's not a corner. You're good.
All right. So, all right, guys, bacteria. Talk to me. The bacterial cell, at its most basic, tends to look like this. There are variations, and we're going to talk about that. But the components of it, in some ways, are very similar to what we see in animal cells, and in other ways, they're rather different than what we see in most animal cells. One jumps out at us. The first thing that will jump out at you is that the plasma membrane is not the outermost layer of a typical bacterial cell. Right? So on our cells, typically, it's okay. On our cells, typically, like we draw the cell, and that's the membrane, right? But in this type of cell, the membrane is found deeper underneath. So what's going on there? You see, the thing with you is, say one of your skin cells, all right, the membrane is the superficial part of it, and if your cells get damaged, you are made of... You made a lot of cells, right? So you lose a skin cell, you're good, you're cool, right? Like you're all right. On the other hand, this bacterium is just itself. It's just made of one cell. So it has evolved basically protective layers. So the capsule is an outer protective layer. That surrounds the cell. And in fact, inside of that is yet another component we're not used to, which is the cell wall, which we sort of think of the cell wall as being composed of like these layers here. So the cell wall includes sort of this outer membrane portion. So this is the total protective layer that surrounds the cell. You'll see here how it says peptidoglycan layer. We're going to get to that in a minute. But we'll say for now, including a peptidoglycan layer. You don't need to know about this yet. We're going to get more into like the molecules of life if you take grade 12 biology, which I hope you will. It's a great course. Peptide should tell you that there's a peptide component to it, which means a protein component. And then glyco should tell you that there's a sugar component to it. Peptido or peptidoglycan. And it's spelled right there. So that is just all protective. All right? One of the things you'll note is that it has all these ribosomes. Do you remember talking about the ribosome in grade 10? Do you remember what it does? I don't remember what they do. Yeah. Yep. Yeah, that's actually really good. That's great. And I heard a couple other people say proteins too. Here's what they specifically do. These read the DNA or the RNA and build proteins from the code. Now, in our cells, there are some There are some free-floating ribosomes in human cells, but most ribosomes in human cells are attached to an organelle. Does anyone remember which one? What organelle are the ribosomes found on in? No, but that's it, the endoplasmic reticulum. And where's the endoplasmic reticulum found? Around the nucleus. Okay, so if you think about it, in our cells, this makes perfect sense. Where's the DNA kept? In the nucleus. Around the nucleus is another organelle called the endoplasmic reticulum covered in ribosomes. And their job is to read the DNA, which is found where again? Yeah, and there's a whole mechanism that takes a small copy of just a section of the DNA we want and moves it out into the ER, where the ribosomes will read that and build a protein from it. And then, because our cells are complicated, we'll actually build a protein from the DNA, and then in the ER, that'll get modified depending on what type of job it's going to do. We have like a whole factory system going on, and then it gets shipped out into the cell. But bacteria don't have any of that because they are prokaryotic, which means they have no organelles at all. So for them, the ER is just floating around in the cell. And in fact, I forgot to mention it here, but we call, does anyone know what we call the arrangement of, well, actually it comes up later. Anyway, we'll talk about what we call the arrangement of DNA or RNA in a cell like this. Now, some bacteria have pili. These are small projections used to bind to other cells or surfaces. All right, so if a bacterium wants to stick onto something, it's got these projections that can be attracted to and can bind to certain other types of things. What's this guy here? Flagellum. What's that for? Yeah, for motion, for movement, right? This is used for motion. Not every bacterium has a flagellum. Not every bacterial flagellum is the same, but it's a structure we see quite frequently. Most animal cells, of course, don't have flagella. Of course, one that we conspicuously think of that does, right? Human spermatozoa, right? That have to move because they're a rare case of an animal cell that works in a single-celled kind of fashion, right? That has to get around and move around and sort of in controlled motion. Most animal cells are busy being stuck together, and we do our moving not cell by cell, but with our whole organism, right? Like our skeleton gets pulled around. Other characteristics, well, like we said, they're always single-celled. They're prokaryotic. The DNA or RNA is in a loop called a plasmid in the cytoplasm. So it's got no protection except the protection of the cell itself. Whereas in our cells, what protects the DNA? Yeah, the cell membrane, but also a second membrane, which is the membrane of the nucleus. Very good, both of them. And what do we call the nucleus's membrane? The actual name for the cell membrane, we call it the cell membrane. That's the membrane of the cell. What do we call the nucleus's membrane? What's the adjectival form of nucleus? Sorry? That's the plural of nucleus, absolutely. Very good. And it's a weird one, right? That double I. But what's the adjectival form? Like this is the, very good, the nuclear membrane, right? Nuclear refers to the nucleus. So the nucleus's membrane is called the nuclear membrane because nucleus is weird to say. Okay. Now, what do they look like? Well, in this particular case, it's got this almost pill shape to it. And that is one of the shapes we see typically, called a bacillus shape. But we also will find what we call the coccus shape, which is these spherical shaped cells. And we'll see what we're called these spirillum shape, which are these sort of spiral shaped. So bacteria can have many different shapes. We also define them based on how they are grouped together. So bacteria, I should say rather, are grouped by their shape and arrangement. And you've probably heard this before. Have you ever had strep throat? strep throat is actually a disease caused by a particular bacterium. Now, there's millions of bacteria that arrange, well, probably not millions, but there's thousands of bacteria that arrange this way, but by a streptococcus bacteria, which means a long chain of spherical bacteria. Does that make sense? So streptococcus would refer to a chain of spherical coccus-shaped bacteria. On the other hand, if you have a staphylococcus infection, that is, again, these spherical-shaped bacteria, but arranged in these kind of clump patterns as opposed to as a long chain. Does that make sense? Sorry? Yeah, but then you could also have a streptobacillus infection. Does that make sense? And in that case, that would be, what does the strepto portion of it tell us? That they're arranged in a chain. And the bacillus would tell us that they are rod-shaped. Yeah, that kind of capsule, kind of pill-shaped. You could have a diplobacillus arrangement, which would mean that they pair up. Does that make sense? So I don't want to have to write 52 examples, but yeah. So that's one of the main ways we group them. And again, that does not mean that every streptococcus bacterium is the same bacterium. They're not. Okay, there's thousands of bacteria that arrange themselves that way. But it's kind of like what we do with animals, right? Where we say all the ones that have a spinal cord, we call chordates, right? We put them in a group together. That doesn't mean that you're the same thing as a giraffe. Well, I'm on giraffes today, but I am. It just means that you have that in common. Does that make sense? All giraffes are chordates, but all chordates are not giraffes. Likewise, you can have a lot more than one type of Staphylococcus bacteria, but that tells you that's one of the ways we categorize it. Oh, this bacterium we're looking at right now, well, one of the things we know about it is that the cells are rod-shaped and that they form in these kind of clumps. Okay, that at least gives us some information about what type of bacterium it might be. Does that make sense? Is that enough information to identify the species? Not by a long shot. It just gives us some information. So what's some other information we could get? Well, as it happens, there's an evolutionary characteristic that roughly half of bacteria have and roughly half don't. And it's something we call gram staining. And basically, if you take a look here, this is more detail than you really need to know in high detail, except to say that we said that every bacterium has a cell wall. Well, some of them have a cell wall where this molecule peptidoglycan is the outermost surface. All right, so in this case, and I should write it below, the outermost surface is this material, this molecule called peptidoglycan. Peptidoglycan. Whereas here, the outermost surface is a material called liposaccharides. Just to break this down, peptido refers to peptides, which are protein-based, and glycan refers to glycogen, which generally speaking is sugars. All right? And just so we know the words, what does lipo or lipo as a prefix refer to? What type of material? Lipase is that's an enzyme that breaks down lipose type structures. It refers to fats. All right? Yeah, lipids. That's right. Lipids. Very good. And then saccharide actually also refers to sugars. Here we've used glycan, and here we've used saccharide. But basically, I just want you to know that these names don't just mean nothing. Like, they describe what these molecules are. But we have this material called gram stain. And I actually have always meant to look up if it's named after a person. I assume it's named after a person, right? It seems like a weird name if it's not named after a person, but I've never looked it up. But basically, gram staining sticks to peptidoglycan, but it doesn't stick to liposaccharide. So gram stain, and when I say staining, I literally mean like a stain. What's a stain? Anyone ever do woodworking? When you're at the staining stage, what do? Usually you take like a sponge or a cloth, maybe a brush, and you dip it in like a can of liquid, and you brush it on the wood, and then you actually wipe it off. Like you wipe all of it off, but the pigment makes its way into the wood and it changes its color, right? So it's weird. You like wipe it on, you let it sit for like 5 minutes, and then you actually wipe it completely dry, but the stain has made its way into the wood and it changes its color, right? And if you do that 3, 4, 5 times, you can get how much color you want. If you tried to use that same stain on like a piece of glass, it wouldn't stay. Like it might stick a little bit, but it would wash right off. Whereas once the stain's in that wood, it won't wash off. Does that make sense? So this stain sticks to peptidoglycan, which means only gram-positive cells, the ones where the peptidoglycan is the outer surface, will change color when stained. Now again, on its own, does this tell us what the species of bacterium is? No, right? That's just one little piece of information, but it does cut the number almost perfectly in half. Does that make sense? Like half of bacteria won't respond to this stain, they won't pick it up, and half of them will. So if you're trying to figure out what bacterium you're looking at, if you do a gram stain and see if it picks up the color or not, you've immediately eliminated about half of the possibilities. Does that make sense? So for something that's small and microscopic, if you look and see what shape it is, how they clump together, and whether it takes gram stain or not, you're narrowing down what type of cell you might be looking at. Does that make sense? It's harder to tell bacteria apart. With animals, they're so big we can look at lots of stuff, right? Cats and dogs are actually really similar, right? They have a lot in common. And if you just looked at them like maybe the way little kids draw things, you probably couldn't tell the difference, right? They both have 4 legs and a tail, and they walk on all 4 legs. You know what I mean? But you're able to pick up details, and slowly the more details you pick up, the more you're able to tell the difference between them, right? And really, like, if you actually tried to put into words, what makes a cat different than a dog? Like, you have to start talking about very specific, sophisticated stuff, right? They both have 4 legs, both have tails, so what is the difference? They both have fur? They make different sounds. Good, right? Mind you, lots of dogs make different sounds too. But how? Yeah, you can talk about snouts and stuff. And again, I don't want to go down that rabbit hole because this is why all my lessons last 75 minutes when they should last like 11. But the point is, just think about how many details you have to talk about to really tell the difference between a dog and a cat. And then think, with bacteria, you need that same amount of detail, only they're tiny. And if you've ever looked at cells under a microscope, easy to see lots of detail. You can see all the stuff inside and they're really specific. Yeah, not when I do it. When I do it, they look like little bubbles, right? And you can kind of see maybe some dots and like squiggly stuff in there, right? It's hard to see. So as we can get information, it helps us figure out what we might be looking at. Does that make sense? So don't think that there's something like super magical about gram staining and that's like an important property of cells that like, oh, that's a thing they do. It's just like, no, this is a way we can tell them apart. Roughly half of bacteria react to this stain because on their outermost surface is this peptidoglycan, and then roughly the other half of them don't. It's almost equivalent of saying it's the difference between an animal that has scales and an animal that has fur. Does it tell you what species you're looking at? No, but it tells you what species you aren't looking at. If you look at an animal with scales, is it a cat? No. Is it a dog? No. Could it be a lizard? Maybe. Could it be a gecko? Possibly. Could it be, what else has scales? Snakes? Could it be help me out here, scaled animals? Fish. Sure, right? And then we look for other traits, like its shape, right? Like how they hang out together. Okay, so there are obvious, sorry?
What are gram-positive?
Gram-positive? Again, it's just roughly half of bacteria we call gram-positive bacteria, and these are just ones where the way they've evolved, that their cell wall is the outermost surface of it is this material peptidoglycan. Gram-negative bacteria also have a peptidoglycan layer, tends to be thinner, but they have evolved to have another layer on top of that, this layer that is made of something we call liposaccharides. And again, you don't have to worry about like how that changes the cell's properties or anything. It mostly doesn't. Again, right, like difference between having scales and having skin, yeah, there's some difference in, one's tougher than the other or whatever, but mostly they both do the same job, which is they like keep the inside parts inside and the outside stuff outside, right? And that's the same thing the cell walls are doing here. It's just a way to tell the difference between them. Did that answer make any sense at all? Are you just saying that to shut me up? No. All right. I think she was saying it just to shut me up. Anyway, we'll move on. All right, how do they move? Well, we said already they can have flagella. Flagellum is singular. The plural is flagella. And these are tail-like structures. At one end. Of the cell. Sometimes it has more than one flagellum on the cell, but they're still all coming from the same end of the cell. Does that make sense? We don't see bacteria that have like flagella on this side and then if they want to go in reverse, they've got them on that side. At least not that I've ever seen, that I'm aware of. And if you look, it's actually quite interesting how they are sort of connected to, how they are attached to the cell itself. They aren't just attached to the cell wall. They make their way right into the plasma membrane, the cell membrane itself, where they're connected. We mentioned, we didn't mention cilia. Cilia are similar to flagella, but all over the surface of the cell and much shorter. That plus sign just looks like it was shorted. No. That's the ampersand. Look at that. All right, so if you look at a bacterial cell like that, it would have these kind of ciliations like this, and they could all be used to move. And this would allow motion in more than just one direction, but not nearly as fast motion, right? The flagellum, this big long tail, can really do quite a bit of moving. The cilia, the single of which would be I don't know actually, cilium, I guess. Never thought of it. And then the pili look similar to cilia. They're smaller. But we already said what they're for. What do? They're not actually for motion, so I don't know why I put them in the motion section. Their purpose is sort of latching onto surfaces. Sorry? The same basic material, yeah. Though the difference with them is that they would have, because what they don't latch on by like clamping on. Like they don't have hands. So rather what they would have is chains of molecules that are meant to interact with molecules on other types of materials, like typically other cells, that they bind to. So basically binding sites. Did that answer your question? Yes, that's right. Yeah. You could see page 1 if you want to. And again, the presence or absence of these things would also help us classify what type of cell we're looking at. So the presence or absence of these of course, bacteria, like everything else, what is the easiest, best way to classify them? Well, I shouldn't say easiest. It's actually not the easiest at all, but it's the best way. It's the most effective way. It's the most modern way. Looking at the DNA. That's right. And it's comparing how similar and how different the DNA is. Looking at the genetic relatedness of the organism. Should I write in bubble letters? Should I like... Oh, I'm so messy. I'm such a terrible feel like this would take me a long time. So the difference here is, remember, let's not even look at the labels. In this case, we're looking at the cell wall here, and what is this material that always is demonstrated as being brown? Peptidoglycan. And in this case, we have a peptidoglycan layer, but then there is another layer of membrane. There's another phospholipid layer beyond it. Which one of these cells is gram-positive? Which one of these would react to gram staining? Yeah, that's right. This one here, right? Because the peptidoglycan is the outermost layer. So it's just showing how a flagellum attaches in a gram-positive bacterium and how the way it's attached to the cell of a gram-negative bacterium is a little bit different. That's all. And again, there's nothing special about being gram-positive or gram-negative, just like there's nothing especially special about having skin versus having scales. It's just two different ways that bodies in animals can be covered, and this is just two different ways that bacterial cells can be covered. Did that answer your question? All right. Okay. There's some other classifications. And what I'm going to do is I'm going to leave these for you to fill out. You're welcome. No, it's okay. I'm actually trying to do you a favor because I don't know about you, but I find the sound of my voice horrifyingly obnoxious. And thank you. I know I gathered that. And 75 minutes of it is a lot. But the other thing is, just like I mentioned with everything else, I can't stress enough, when you go to fill this out, I want you to look these up. And I'm working on getting textbooks for us. It's challenging because our education system's ridiculous. I will post a section of textbook, but these are also things you could Google. But I got to stress something for you, which is the difference between getting work done and learning. And I am never interested in you getting work done, which is to say, if I give you an assignment and you haven't learned something by the end of it, then I've wasted your time. Like really, fundamentally. If I give you an assignment and what you do is you go Google a bunch of stuff and put it in there, and a week later, if I asked you about that stuff, you don't know about it, then I am guilty of a horrible thing. Because I've just made you waste time writing stuff down that already was written down by somebody else somewhere else. And spoiler alert, I know the answers to these. So it wasn't to tell me anything, because I know this stuff. So when I give you something like this and say fill it out, I don't actually care that it's filled out for the sake of being filled out. Like the climate crisis isn't going to solve itself because everyone in my class wrote down what aerobic respiration is. And like the Israel-Palestine conflict isn't going to be solved because everyone got obligate and facultative anaerobes described. You know what I mean? Like that's a meaningless act in and of itself. Rather, what I want you to do is learn it, right? So this is so important to me because I think so much, and tell me if I'm wrong, I feel like so much of your education has been spent Googling things and then writing answers down and doing as little thinking about that along the way as humanly possible. And I'm not trying to be critical, but is that fair? Yeah. That's a horrible waste of your time, and I hate to waste your time. I respect your time. Like you only get so many spins around the sun in this life, and I really would hate to think that you spend a meaningful portion of any of them wasting your time writing stuff down just for the sake of moving your hands in my class, or typing, or control-seeing, and control-being. But I think there's a lot of value in learning things. I think learning stuff is one of the great things humans do. Like the more stuff you cram into that brain of yours, the more useful your brain is. It's just the way it works. You find me one person in the world who you think of as really wise and you think of as really smart and you really trust who doesn't know a lot of stuff, right? Like I go and ask my dad for advice on stuff because my dad knows a lot of stuff. And I go and ask my mom for advice on different stuff because a lot of stuff my dad doesn't know. My mom's a much smarter person to ask advice, right? But like in the end, the people who are wise and the people that they've crammed a bunch of stuff in their brain, and then once all that stuff's crammed in your brain, you can connect it to other stuff, you know? And like once you cram a bunch of math into your brain, one day you'll have a job managing a restaurant, hypothetically speaking, and go like, hey, I bet I could write an equation that could help me figure out, based on how many reservations we have and the temperature and what day of the week it is and all this other stuff, how many steaks we should unthaw, because we keep unthawing, not unthawing, that's not even a word, thawing, we keep wasting too many, you know what I mean? And you do this and all of a sudden you cut your waste down by 15% and the place is making money and then you get a raise. Nobody asked me to do that. And nobody told me to do that. Nobody told me I could do that, which means that the fact that it was a Googleable thing wouldn't have helped me because the only reason I thought to do that is because I'd crammed my brain full of a bunch of restaurant knowledge and I'd crammed my brain full of a bunch of math knowledge. And then this amazing thing happens. When you cram your brain full of stuff, they start talking to each other. And they start saying, ooh, hey, we know this thing, but we know this other thing. And when you go to history class and you annoyingly memorize the order of the presidents of the United States and the prime ministers of Canada and what date this happened, what date that happened, now this amazing thing happens where when somebody starts telling you about the history of this country over here that you know nothing about, in your mind you already know what was happening in the rest of the world at that time. And it's going to make you understand this thing better. Again, yeah, could you Google all that? Of course you could, but you wouldn't. You wouldn't think to, right? But when you cram a bunch of stuff into your brain, the things in your brain talk to each other, all right? So when I give you something like this and say, go get the answers on here, if all you're going to do is copy and paste them, just don't. It'll have the same effect on your learning. Do you know what I mean? And at least then if I look at your notes, I'll know that you don't know it, right? Like, just don't. Like, don't waste your time. That's a waste of your time, so don't waste it. I don't want to waste your time. What I do want you to do is look these things up, read them, and you're not done until you're like, oh, yeah, I get it. Oh, I know what that means. And then, since you know what it means, put away whatever you were looking at, put away the Google article, put away the YouTube video, whatever you watched. The resource doesn't matter. I don't care where you get your information. Put away the textbook section that I put online. And then here, in your own words, tell me what it is. Does that make sense? That's useful. That's not wasting your time. Does that make sense? Cool. Yeah.
Are we doing this now, or is it homework we're looking at?
You'll do it in the very little bit of time left. So just this one section here. Other classifications, I'm going to let you fill these out on your own. Who is calling me? I'm a high school teacher. Honest to God. Okay, sorry, pardon me. So, other things we look at. Honest to God. Oh my goodness, I am so sorry. Like, does it just say God? Does this happen to you? This has never happened to me.
Was it your wife?
Well, then I would have said it was God, right? No, it was not my wife. It was Eden High School, so I'm assuming, like, my daughter is horribly injured to stay home to skip school or something. I don't know. I'll figure it out after school. Anyway, classifications by feeding habits. This one we will fill in together, because this one I think we can break down. What's an autotroph versus a heterotroph versus a chemotroph? First of all, what do all of these contain as a suffix? Troph. Well, troph, which does not have an H at the beginning, refers to feeding. All right. So what does auto mean? Automatic. Now that's interesting because an automobiles. What's it mean to be mobile? To move. So something that's automobile moves how? Autoly. What does auto mean? On its own. An automobile moves by itself. You don't need horses. See, an automobile looks an awful lot like a wagon that people used to drive. The difference is with a wagon, you had to get a couple of horses and strap them to it and be like, hey, you guys, go. And then the thing moved. But a car, it moves itself. It's an automobile. So an autotroph does what? Very good. So these can produce their own energy molecules. And the most common one we think of is what plants do, which is what? Yeah, for example, photosynthesis makes sugars. Of course, what does it need to make sugars? It needs sunlight. What else does it need? Water. What else does it need? Sorry? Yeah, very good. Right? What's a heterotroph? If autotrophs feed themselves or create their own food, heterotrophs, what does hetero mean? Opposite. Yeah, other or opposite. And in this case, we're going to go other, right? Other feeding. This is what we do. We get energy by consuming other living things. Right? By the way, where did the other living things get their energy? Well, if they were heterotrophs, they got it by eating something else. We call that a food chain, right? But eventually, if you go far enough down the food chain, what do you get to? An autotroph. Something had to make the energy, right? Think about your life. You have never once made a piece of energy. Rather, you find other living things enjoying their happy little lives. And by the way, all you vegetarians who are so smug, I got news for you. You also kill. You just kill plants. Which I guess is better because to the best of our knowledge, plants don't have like feelings or emotions or like pain senses. But even still, you're eating an apple. I got news for you. That apple was happily sitting up on that tree one day, just swinging around in the breeze, probably enjoying the sunshine, being like, hey, you know what's nice? Being on this tree right now. You know what would be less nice? Being ripped apart by somebody's molars. Next thing they know, they're in a brown paper bag in some kid's backpack, spending all day in a smelly locker, and then get chomped on by like a kid named Charlie's molars, hypothetically speaking. Yeah. Right? But the point being, what? I was thinking of my Charlie. But anyway, I have no idea if you put an apple in your lunch. Do you bring an apple to school? I love apples. It's one of my favorite foods. Anyway, but so heterotrophs have to get their food by consuming other living things. And again, every single molecule in your body you got by killing some other living thing out there and shoving it into your face. And neonatally, like your mother shoved living things into her face and just ripping them apart with your teeth and grinding them down with enzymes, putting them in your stomach where acids rip them apart. And then like also the stomach like churns and grinds them and then absorbing them into your bloodstream. And that's how you make stuff. It would be like if you wanted to build a house, the way you did it was going around your neighborhood, stealing supplies off other people's homes. Be like, oh, you know what? I need bricks. Like going and like kicking them off your neighbor's house and be like, these are my bricks now. That's the only way. Sorry? I could. I'm pretty tough, but I don't as a rule, no. Okay. And then what's a chemotroph? Yeah, these are able to make energy molecules. And when I say energy molecules, these are molecules the cell uses to store energy. So they're able to make energy molecules. The most common example is sugars, but there's others. To make energy molecules from the energy released by chemical reactions. So, for example, there are archaea that live at the bottom, like kilometers underwater, where no sunlight gets down there whatsoever, and they live in like these geysers where like there are underwater volcanic eruptions happening, and they actually thrive there and they do well, and that's because they're chemotrophs. They're actually able to take the energy of the chemical reactions that are happening down there because of all that heat and all that pressure, and they're able to use that energy to provide energy for themselves. You would not do well living in an underwater volcano, but they do just fine. I think I'd be all right. I'm pretty tough. First thing people know about me. Okay. No. No. Most bacteria reproduce using a process called binary fission. Keep in mind that the mitosis, the process you learned about in grade 10 science, is in some ways way more complicated. Because if you think about mitosis, what are you immediately talking about? So the first step. What's the first step of mitosis? What's the first step of mitosis? Yeah. Your toes just exist, but mitosis, a little pun. Kelford would have enjoyed that. He would have. Interphase, sure. But once we get to mitosis, what's the first stage? No, that's the last stage. Okay, what is it? Prophase, good. What happens in prophase? Do you remember? They actually have already replicated their DNA. That happens during interphase. What happens during prophase? Okay, and for it to move away, what has to go away? Not the cell membrane, the nuclear membrane. Good. So in prophase, we're immediately talking about nuclei and chromosomes because we're talking about complicated cells, right? Of course, bacteria, way simpler than that, right? So bacteria, actually, you got a happy little bacterial cell here, and maybe it's got flagella. I'm going to have to draw that every time then, and I don't want to do that. All right. And so it's leading its happy little life, and it's got its DNA or its RNA there. Does anyone remember what we call this loop of DNA or RNA? The plasmid, which is just its DNA or its RNA. And then when it's ready to reproduce, it does two things. When it's ready to reproduce, and bacteria frequently, basically, that's all they do. They kind of get born and then they live a little bit, then they reproduce, then repeat. Two things happen. One is that the cell is going to start to grow bigger. All right, so the cell enlarges. And then secondly is that plasmid is going to get duplicated. We're going to make a copy. And I'm just showing them in two colors so it's more clearly two copies. Does that make sense? But the other thing is the DNA or the RNA is duplicated. Then they are going to start to pinch off in the middle like that. And of course, for that to happen, we want one copy of the DNA to go to this side, and we want another copy to go to that side, just like that. And then what's going to happen? They're going to fully separate. And what do you have? Two daughter cells. How do they compare to the first?
They're smaller.
Sorry? There are definitely more of them. Yeah, I mean, eventually they're going to grow, right? So they'll grow to that side. But for sure, more fundamentally, this is obviously a different mechanism by which we reproduce. But the other thing is when you reproduce, so if one day you choose to reproduce and you have offspring, what will your offspring be like? Well, you don't know. Why don't you know? That's why they're their own thing, right? They incorporate your DNA, but A, they also incorporate somebody else's DNA. But secondly, there's a lot of randomness that goes on to which DNA they incorporate and which they don't. And so they're their own unique combination. Is that true for these bacteria? No. So here we have two daughter cells that are clones of the original. And in fact, it's conservative, which is to say, it's semi-conservative, which is to say one of these actually just has the original piece of DNA. Does that make sense? But the other one has a copy. I'm sure they do, but we're not going to write them down. Now, bacteria can, and from time to time do reproduce sexually, although their form of sexual reproduction is also quite simple relative to ours. What happens when they do reproduce sexually is that you can get basically what is called an F plasmid, and that sounds fancy, but basically there's just some bit of DNA that can be present in one cell, and what can happen is that these two cells can come together, and when we say F positive and F negative, that sounds really fancy. All it means is this cell currently has this little bit of DNA. And I wish that this showed this better. This is basically usually like a copy of something that's already in here. Does that make sense? So this little piece of DNA can get copied. And then when these two cells connect, what can happen? Or actually, sorry, no, the way they've shown it here is different. They're showing it as a separate piece. That's fine. And what can happen is that this duplicates itself. Do you see that? How it's double-stranded? Like two copies? And what can happen is that one of the copies can get transferred from one cell to another. Do we kind of see that here? And the result is that at the beginning, one cell had some piece of DNA, while the other cell didn't, right? And when they come together, a copy of it gets transferred across, such that when we're done, both cells have that copy of that piece of DNA. Does that make sense? Bacteria will naturally do this under certain conditions. So bacteria often stick together. What structures do they use to do so? When they do, they often transfer copies of segments of the DNA from one to the other. And this can lead to the transfer of traits. So this is very different. Here's what doesn't happen. When humans and other animals and plants engage in sexual reproduction, you don't take on the genes of your partner, right? It's not like you have like sexual intercourse with somebody and then all of a sudden you have like their hair color, their eye color or something, right? That's not the way our sexual reproductive process works. You're not taking their DNA and incorporating it into our own. In our sexual reproductive system, we take a portion of our DNA, they take a portion of their DNA, we put them together, and we start a new cell. If you look here, bacteria have a very different method where they literally just take in the traits of the other cell that they are, sexually conjugating with. So it's a very different mechanism and really a much simpler mechanism than what happens with us. So this can lead to new DNA for one of the cells, which means new traits. This is also dangerous. This means that if we have an antibiotic we use and some bacterium out there develops an immunity to it so that one of our antibiotics no longer works, it will randomly from time to time conjoin with other bacteria and pass along DNA, and there's a possibility that it can pass along that gene. What happens if a bacterium that isn't killed by our antibiotics happens to bump into a bacterium that poisons and kills us and transfers that along? Well, now you have a bacterium that poisons and kills us that also has a gene that helps it not get killed by our antibiotics. Pretty dangerous, right? This, by the way, happens every day. This is another thing to know. Like 10 years ago, everyone was saying, hey, a global pandemic will happen someday soon and we should be ready for it, and we weren't. There's another thing you should all know, because it's the truth. All of the antibiotics we currently use will in the near to semi-near future stop working. Because there are bacteria out there already that are resistant to them, and that resistance spreads more year by year by year because bacteria talk to each other sexually in this way and pass along traits. And so a few things are true. One, the more we use our antibiotics, the more we encourage if you think about it, if you kill every bacterium in your body except the ones that are resistant to your antibiotics, well then what bacteria are left to reproduce? Only the ones that are immune to our antibiotics. Then those bacteria start talking to and hanging out with other bacteria. They spread that along. When was the last time we developed a new antibiotic? The 1960s. Why don't we do it anymore? There's no money in it. There's no money in it, right? Because for drug companies, they're always trying to find a new research and development of a drug is incredibly expensive. And how do they pay for it? Well, if we were smart, the government would pay for it, and we'd say, hey, pharmaceutical research is really important. It saves lives. We should invest money in this. We don't do that. No. We let the private sector take care of it in for-profit businesses, right? But when for-profit businesses do it for us, there's no incentive for them to develop drugs that do things that we already have drugs for. Does that make sense? Why would they? Where's the money in that? We're going to spend billions of dollars and develop a drug. Who's going to take it when it doesn't work any better than anything else? So for right now, there's no reason for pharmaceutical companies to be developing new antibiotics. So they don't. The problem is, again, we know that in time, the ones we currently have will stop working. Kind of a scary thing. And every year you hear about more and more outbreaks of dangerous bacteria in hospitals that are antibiotic resistant. And what are we doing about it right now? Nothing, because that's what we're really good at. We like to wait until there's a crisis, and then we like to do as little as possible to solve it once we're in crisis mode. And that is still incredibly expensive and disruptive. Look at COVID, right? This thing came and hit, and we had to shut down the whole friggin' world, whereas those things that can be done in advance, preparatory, and are a lot cheaper and a lot less disruptive, but because nothing bad is happening yet, we go, well, why would we spend money on that? When we could spend money on, I don't know, the Super Bowl and guitar pedals. I like to pick on it, whatever. But why spend money on doing something that doesn't solve a problem we have right now? Oh, because it might solve a problem in the future? Well, I don't live in the future. I live right. You see what I mean? People, not very smart. And I don't mean other people. Mostly I mean me. But it's, when your generation gets to voting age and gets to be the ones who are out there running the world, my advice would be smarter than my generation, because we're dumb. My generation might actually be the dumbest generation, really, if I think about it. I think my parents' generation, and they were at least out there protesting wars and fighting for women's rights and stuff. My generation is a generation that said what if we let women's rights backslide horribly so that women in 2024 have fewer rights than they had in the 80s? You see what I'm saying? My generation really sucks. But you guys will be the next generation, and you have the option of not sucking. And you know what I would recommend? Be cool. Go out there and do good things and make smart decisions and vote for people who say, hey, we'd like to solve problems proactively. And If politicians don't want to solve problems, go like yell at them and like stand outside their offices and hold signs that say like, hey, we'll vote for the other guy if you don't start doing something. You see what I'm saying? Because my generation is too busy being like, yeah, I'd like to go to Starbucks. Everything's fine. Of course, you should like Starbucks. But like go to Starbucks on your way to going to do something like useful for future you. What might be? Why is that? Oh, interesting. I did not know that. Interesting. Well, I love controversial topics, but my goal is to be done 30 minutes ago. So, last thing, another way that cells, and we don't even have to write anything here, just something to note, bacteria themselves have short lifespans. But they have the ability to lie dormant for potentially very long periods of time by forming something called spores. So they occasionally can form spores, which can lie dormant for long periods of time. But then can come back. This is not a course in like microbiology where we have time to dig into all this stuff. You do not need to know the specifics of this diagram. If you did, I would have written it down in words. Does that make sense? But I just want you to get the idea that bacteria themselves have some reproductive methods that go beyond just dividing. They have a simple form of sexual reproduction wherein they can transfer genetic material. They have a version of reproduction that allows them to sort of survive difficult conditions, right? Because spores are very tough in ways that bacteria themselves are not. They have what's called the exosporium and the spore coat, all of which is to say they have this big protective layer. Okay, I took too long, as always. I'm sorry. I think I need to start like challenging myself where I say like, if I take too long today,